Okay, thanks, Uli. Thanks, Uli. And before I start, I'd like to thank you for the chance to talk and organizing the seminar, the conference. It's been great. So this project I'm going to be discussing was a collaboration between my lab at Scripps and the labs at Georgia Tech of Lauren Williams and Nick Hudd. And this was done as part of the Center for Chemical Evolution. So my presentation is going to focus on a project exploring interactions between nucleic acids and protopeptides, and I'll describe more what I mean by that, and how those interactions might have helped mutually promote the emergence of both nucleic acids and peptides in the origin of life. So as we all know, interactions between proteins and RNA are everywhere in biology today, and my favorite example of that is the ribosome, which is this cool molecular machine that's composed of many proteins and RNAs all working together. So shown here, RNA is in red and proteins are in blue, and you can see how they fit together like pieces of a jigsaw puzzle. And this is what the partnership between RNA and proteins looks like after billions of years of coevolution. And our question is more in what the partnership might have looked like at the very beginning during messier prebiotic environments at the origin of life, right? It seems certain that at some point in the origin of life, there were productive interactions between nucleic acids and peptides that prompted the evolution to the current RNA, protein, DNA system that we know today. But how did this interdependence come about? So the question we set out to answer was, can there be functionally meaningful interactions between nucleic acids and very simple non-coded protopeptide oligomers that are produced in prebiotically plausible reactions? Okay, so in practice, how do you go about answering that question? Where do you start? And our starting place was to generate and characterize a collection of protopeptides for our studies. So what is a protopeptide? For us, that means short oligomers that are produced in wet-dry cycling reactions involving amino acids and hydroxy acids. So these reactions produce random condensations between the monomers that are present to give peptides and depsipeptides. Depsipeptides are just a name for peptides that have an ester bond in place of some of the amide bonds in the backbone. So compared to what Burkhardt just presented, these oligomers are much shorter, usually less than 10 residues, and they're non-coded. And these really are mixtures. There are different lengths of oligomers present, shorter ones and longer ones. And for our studies, we wanted to generate cationic protopeptides. And this goes to Nita's question to Burkhardt, ones containing positively charged side chains, because we thought this would favor interactions with nucleic acids through attractive electrostatic interactions. So we definitely cherry-picked and chose cationic amino acids, to use Burkhardt's language. So we investigated the small collection of cationic amino acids and hydroxy acids that are shown here. Now, the question of which amino acids would have been present on the prebiotic Earth, I would say, is unresolved. And so we took an agnostic approach. We chose all three of the proteinogenic cationic amino acids. Those are lysine, arginine, and histidine. And we also studied three shorter chain cationic amino acids that have been discussed as potentially more prebiotically plausible or have been found in meteorites or model prebiotic reactions. And we also studied the cationic hydroxy acids that are shown here. Okay, so what do we actually generate when we dry down these oligomers? One complication when you're using basic amino acids or amino acids with a cationic side chain is that you have two potential sites for acylation, right? So you have the alpha amine position, and you also have the side chain amine that can be acylated in these random condensation reactions. Now, if the reaction proceeds with acylation only at the alpha amine, you generate a structure like this, which is what we know as the canonical topology of proteins in biology today. But because these reactions are random, you can also generate acylation at the side chain, or at the side chain and at the alpha amine, which will give you branch structures. So the potential is for very messy mixtures with lots of different products. And at the outset, I would say I was not very confident that our mixtures would generate oligomers containing much or any free amine side chains. You know, I expected a lot of branching, and essentially that most of the amines would be acylated. And I thought maybe we could play around with the conditions or the stoichiometry to try to favor the formation of molecules that were cationic. So our HPLC and mass spec told us that we were forming oligomers, but it didn't really tell us anything about the structure. And so we turned to NMR for that. And on this slide, I'm summarizing a lot of work by Moran, Frankel-Pinter, and Martin C. and other people in the group. But what we found was quite striking. So the three proteinogenic amino acids, lysine, arginine, and histidine, all reacted more efficiently and more selectively to generate what I'm calling the protein-like topology, where you have acylation at the alpha amine and a free side chain. They were much more efficient and selective compared to the three non-proteinogenic amino acids that we studied. So just shown here is the percentage of amino acids that are present in this topology, where you have the alpha amine acylated and the side chain free. And you can see there's a considerable difference between the proteinogenic and non-proteinogenic. So the prediction based on that result is that if you had a competition reaction with one of the more efficient amino acids and one of the less efficient amino acids, what you would expect is that the more efficient amino acid is incorporated into oligomers, whereas the less efficient one was not. And indeed, that's what we observed in several competition reactions, such as the one shown here between lysine and diaminobutyric acid. So in this reaction, lysine is present in the mixture mainly in the protein-like topology, whereas the diaminobutyric acid is present mainly as these cyclic lactam type structures. So this leads to my first take-home point, which is that certain amino acids are inherently superior to others during non-enzymatic oligomerizations. And these reactivity differences could have been one of the factors that contributed to the selection of certain amino acids for use in biology. We've heard a lot about selection this week, how vast libraries of organics are produced in space and on Earth, and how they were eventually narrowed down through some kind of selection process to what biology uses today. And you know, we think this might be one concrete process by which certain amino acids were selected over others. Now, I want to be clear that there would have been many more amino acids and hydroxy acids and other types of monomers present on the early Earth. And for certain, many factors would have influenced which monomers were eventually selected by biology. But if certain amino acids could not efficiently incorporate into peptides or oligomers, then they would never be available for selection at higher levels, like the level of the oligomer. Okay, so moving on. I showed you that we can produce mixtures of cationic oligomers, and the next question we explored was what effects do those have on RNA folding and stability? So this slide is just summarizing how we set up some of our experiments. So we looked at 4 different RNA structures, 3 duplexes shown here with varying GC contents, and then 1 hairpin-like RNA structure. And on the top of this slide, this is just a schematic of how we set up our dry down reactions that I just described. And I just want to note that For some of our analyses, we used the crude mixtures containing depsipeptides and monomers. But for some of our analyses, we dialyze these mixtures with a molecular weight cutoff to remove unreacted monomers and shorter oligomers, to really convince ourselves that what we were seeing was due to oligomers that were present. Okay, so here's what we observe when we incubate dry down mixtures with RNA. And our readout is the melting temperature of the RNA duplex. This is the temperature at which 50% of the RNA duplex is melted and 50% is still hybridized. So a higher melting temperature corresponds to a more stable folded RNA. And what we found is that some of our mixtures, either the crude dry down mixture or the dialyzed dry down samples, some of them significantly increased the observed melting temperature of the RNA. And this worked for all of the different RNA duplexes that I mentioned, as well as the hairpin. Now, in some sense, this is not too surprising, right? It's well known that cationic oligomers can bind to RNA and other nucleic acids and increase their melting temperatures. We heard about interactions in coacervates, which is one example of that. But what I want to stress is that our molecules are non-coded oligomers present as a relatively complex mixture produced in prebiotically plausible reactions with essentially no optimization of the reaction conditions. So these protopeptides were produced in a single drying step with no condensing agents. So to me, this says that it's not very difficult to generate oligomers that would have interacted functionally with protonucleic acids that might be present, right? If certain amino acids and hydroxy acids are dried together, these sort of results would seem to be a natural consequence. Okay, so protopeptides can increase the stability of RNA folds, but what about the other side of the coin? How are depsipeptides affected by interacting with a nucleic acid? And for this, we examined the effect of interactions with RNA on depsipeptide hydrolytic stability. So unlike canonical peptides, depsipeptides are subject to relatively more rapid hydrolysis because of the ester bonds in the backbone. These can undergo hydrolysis much more rapidly than a peptide bond. So this is our model for depsipeptides interacting with RNA and how that might affect the hydrolytic stability, the rates of breakdown. But I'm getting short on time, so if you'll bear with me, I'll just move to the data. So here's the data for a series of experiments at different concentrations of RNA. So in each of these reactions, we have 25 micromolar depsipeptide, and we're just monitoring over time by HPLC the hydrolysis of that depsipeptide into shorter fragments. So what I'm plotting here is the intact depsipeptide. So as time increases, depsipeptide hydrolyzes. But what we observe is that the presence of RNA drastically reduces the rate of that hydrolysis. And so what I'm showing you here, filled circles are data points. That the curves are a simultaneous fit to the model shown on the left using the program SimFit. And what we get out of this is that the hydrolysis rate of the depsipeptide is around 35-fold slower in the presence of the RNA. And putting this all together, I think this single experiment nicely illustrates the potential of these mutually stabilizing interactions. So in this experiment, we have 3 samples all prepared from the same stock solutions at the same time. One of them is RNA alone, so you can consider this the RNA world. One of them is depsipeptide alone, so a protein world. And then in the third vial, we have both RNA and depsipeptide. And so these samples are incubated for the same amount of time, and then we measure either the extent of RNA hybridization or the hydrolysis of the depsipeptide. And what you see is that with RNA alone, there's there's only a small fraction, about 10%, that's folded. Whereas in the presence of the depsipeptide, because the depsipeptide stabilizes and increases the melting temperature, there's a much greater extent of folded RNA. On the other hand, looking at depsipeptide hydrolysis, in the absence of RNA, the depsipeptide is essentially completely hydrolyzed, whereas in the presence of RNA, you still have the majority remaining. Okay, so all the data on the previous slides were for double-stranded RNA, but what about other nucleic acid structures? So shown here is the rate of depsipeptide hydrolysis for a few different nucleic acids. So we have duplex RNA, duplex DNA, single-stranded RNA. And you can see there are considerable differences. So what about other types of pre-RNA or protonucleic acids that might have been present? And what about other types of protopeptide structures, right? Heterochiral peptides or beta peptides, for example, or take your favorite proto-oligomer. It seems likely that certain types of nucleic acids and certain types of peptides will be better able to interact with each other due to structural compatibility of whatever type of secondary structure they favor to form. And when you consider all the possibilities of different types of protonucleic acids and protopeptides that could have been present, I think it's easy to imagine how selective interactions between certain classes of molecules could help those molecules persist through mechanisms such as this. And that leads to my last slide, which is that mutually stabilizing interactions provide a basis for selection in prebiotic evolution. We know that cooperative interdependent partnerships are a hallmark of biology, and we think that they were crucial during the origin of life as well. So I'll stop there. With that, I'd like to thank the people who contributed directly to the work, whose names are listed here, as well as everyone from the CCE, and especially Moran and Lauren, who made this a really fun project to work on. And I'd be happy to take any questions.